Hello students. Now in this class we will study male accessory ducts and male accessory glands and their functions. After that we will see the process of spermatogenesis, spermiogenesis and the hormonal control of spermatogenesis and spermiogenesis. So to begin with, various male accessory ducts and male accessory glands. Now there is a difference. Male accessory ducts. Now among all the parts which are shown here in the slide, let me label one by one and then we will differentiate. We will see who all are the male accessory ducts and who all are male accessory glands. So you all know that this is a testicular lobule. This is just the revision of the previous slides, that these are the testicular lobules, right? And this is where the rete testis is coming here, then the vas efferentia which carries the sperm from testicular lobule to vas efferentia, and then vas efferentia carry the sperms to the epididymis. So testicular lobules, after that rete testis. These structures are somewhat here. Then the straight few in number tubules, these are actually vasa efferentia. And then these vasa efferentia will carry the sperm to the largest coil tube, which has got three parts. We have already studied in the previous slide, that is the epididymis. Okay, so here is the epididymis. From epididymis, the sperms get the functional maturity in the epididymis, and then they will enter into the tube, a single tube, actually pairs, but one side only one tube. So a tube which is coming out from scrotum, and then it goes upward in abdomen, and then it will take a round around the urinary bladder. This single long tube is known as vas deferens. Now, after taking a loop around the urinary bladder, then this duct will join. Here is the vas deferens, and this vas deferens will join with the another structure which is present here in the pair 1 and 2. One pair, that is 2 in number. This structure is actually seminal vesicle. Now the duct which is coming from seminal vesicle, this duct will be joined with the duct which is coming, that is vas deferens, and then they will form a single duct which is actually ejaculatory duct. It is not, we cannot show here the ejaculatory duct. So that ejaculatory duct will then join with the duct which is coming from this structure, this part you already know, urinary bladder. So we have seen that vas deferens joins with the duct coming from seminal vesicle, forms ejaculatory duct, and ejaculatory duct will then join from the duct which is coming from urinary bladder. At this point, they also join with the single large gland which is present here. This structure is prostate gland. Now, and then these will join the one more structure which is, you can see here, the pea-sized, a pair of structure present here. This is actually Cowper's gland. So the tube which is coming from Cowper's gland, it also opens into the duct which is now, see, so many ducts are joining and they will enter into a single duct which is now we have to label this part, that is urethra. And this duct is actually coming right away from urinary bladder, and the vas deferens first join to the seminal vesicle, it will join this urethra, then the prostate gland starts secreting in this urethra, then the Cowper's gland, again they start releasing their secretion into that urethra. This urethra, now it has entered in the external genitalia, this external genitalia which is actually known as penis. Now the anterior portion of that penis is known as glans penis, which has a skin which is known as foreskin or prepuce. Now, so this is glans penis, and here is the skin which is foreskin. So this labeling is, of course, you all know testis. That's it. So we have seen that testicular lobules then lead to retitestis, vas efferens, epididymis, vas deferens. Okay. Now one more labeling is there, right? Ureter, because the ureter come and open into urinary bladder, right? So this is ureter. So 1, then 2, then 3, and then 4. All these are the ducts. They have, the function to carry sperms, spermatozoa, from testis to the duct which is coming from the urinary bladder. Okay, that is urethra. So they carry the sperms. They have the important function for transferring the sperm from male reproductive organ, the primary sex organ, which these sperms will be carried to the part from where these sperms will be released into the female reproductive tract. So these are actually accessory ducts. Accessory ducts. Actually, they don't have any important secretion, but here the seminal vesicle, which is actually pair, so 2 in number, a single prostate gland, which is 1 in number, and the Cowper's gland, which is again pair. These 3, 1, 2 and 3, seminal vesicle, prostate gland and Cowper's glands, these are known as male accessory glands because their secretions play important role in the formation of the semen, which consists of sperms as well as the chemicals or the materials which are required to make the sperms live, to make the sperms mobile, and also to provide the sperms energy, make to neutralize the acidic effect of the urethra. So all these functions are done by the secretions from these accessory glands. Now one by one we will see the functions of these male accessory glands. So now it is clear for you that what is the difference in male accessory ducts and male accessory glands, right? Now this is a fair slide. Just now we have labeled all the parts you can see here. So male accessory ducts, vas efferens, epididymis, vas deferens and retitestis. They serve to store spermatozoa to transport them outside for ejaculation. That is actually their function. But glands, seminal vesicle, prostate and Cowper's glands have the second name, that is bulbourethral gland. They contribute seminal plasma. Their secretion contribute to the semen, and very important roles they play. So one by one now we will see the functions. Okay, functions of accessory glands. First and most important, that is seminal vesicle. Now seminal vesicle which is, we all know that the largest, this not the largest, but the pair of gland which is present, okay, and this seminal vesicle is actually have 3 layers, right, and the inner layer of that seminal vesicle produce seminal vesicle fluid. That fluid contains seminal plasma, and it actually constitute 70% of the total semen fluid which is ejaculated into the female reproductive tract. So the fluid of that seminal vesicle, so seminal vesicle fluid contributes 70% of the seminal fluid of the semen, 70% of the semen. Okay, now what is the function? Because this contains first fructose. So we all know that fructose is a carbohydrate, and it provides energy, of course, for the movement. The energy which the sperms need to cover the distance from female reproductive tract through the uterus, and then they have to reach to the oviduct for fertilization. So energy is provided by fructose, and very important point here to note that this fructose is not present anywhere in the body, anywhere. Why? Actually, this fructose is present only in semen, the secretion from the body organs, and this can be used for the forensic test for rape cases. So in the victim, if the fructose is found in the victim's reproductive canal, then this rape is confirmed. So it is used, this presence of fructose is used for rape cases. Okay, so because it is not produced anywhere else in the body, so it provides a forensic test for rape. Okay, the second thing which is present in the in that alkaline, this seminal fluid, is it is actually alkaline in nature, so it neutralizes the acidity of the male urethra and also present in female vagina. So alkaline fluid which neutralizes the acidity. Okay, now the next one, second. Now the third one, prostaglandins. Now the prostaglandins which are present, these are actually the hormones like structure. They have a role in lowering the female immune response in the semen. So prostaglandins are also secreted in by seminal vesicle. Please remember that prostaglandins is not secreted by prostate gland, but prostaglandins are secreted by seminal vesicle. And some more things which are present, that is the phosphorus, the potassium ion, vitamin C. Okay, vitamin C is also present, potassium present. So these are which help in mobility also, and which help the sperms to move, right? Help in movement. So all these structure, fructose, alkaline fluid, prostaglandins, potassium, vitamin C, all these are the structures which are present in the fluid which is secreted by seminal vesicle. So there actually it is also provide the movement also, and you can write here nourishment also, so nutrition also. So that's all for seminal vesicle. Now we'll see the function of the second gland. This is prostate gland. Actually the prostate gland is its secretion is slightly acidic, and 20 to 30% the semen is consist of that the secretion which is from prostate gland. Its pH is 6.1 to 6.5, but overall pH of semen becomes alkaline. So there is no effect of the acidic secretion which is from prostate gland. But the function of the prostate gland secretion is actually the secretion, it contains zinc, calcium, then citric acid, then enzymes like phosphatase, then pepsinogen, because its secretion is actually the milky secretion, first one. So you can write here the milky secretion which contains calcium ion, then Zn, then citrate. So that would be providing nutrition, of course, right? And it also help through the enzymes which are secreted, that is fibrinolysin. Okay, now what is the function of this fibrinolysin? Liquid for coagulated semen after its secretion inside the female reproductive tract, so that the sperms will become free and it will reach to the female reproductive tract. Okay, then it has the enzyme phosphatases also. Okay, so the various functions from prostatic fluid, that is, it contains a number of ingredients like several enzymes, zinc, then citric acid. So it is also it helps the sperms to live longer and they will be more mobile. Okay, now we'll see the third accessory gland, that is Cowper's gland. Cowper was the scientist who discovered this gland. So Cowper's gland, it is also known as bulbourethral gland. Of course present in pair. See, the largest gland, which is the prostate gland, it is 1 in number, but the Cowper's gland and seminal vesicle, they are present in pairs. Bulbourethral gland or Cowper's gland, it is actually they are secreted just prior to ejaculation. So it secretes 4 ml, approx 4 ml fluid at the time of sexual arousal, and this is a clear fluid which is rich in mucoproteins. This mucoprotein provides the lubrication so that it can the movement of the sperms will be easier, and it also neutralizes the acidity in the urethra. So it neutralizes the acid effect and it lubricates the pathway. So that's all for the functions of 3 accessory glands. Now we'll move to the next. Here is a slide. We have just now discussed that what are the functions of seminal vesicles. The first one is seminal vesicle, and the functions of seminal vesicle, all is written here. The normal pH of the semen is 7 to 8.3. Okay, so that the semen can neutralize the acidic effect, because the sperms cannot survive in the acidic effect which is present in the urine. Okay, so the pH of the semen is alkaline, so that it can neutralizes the effect. So, and here the prostatic fluid, the functions of prostatic gland and the bulbourethral gland. Okay, now here is a question for you. Prostaglandins are secreted by which gland? So don't get confused. Prostaglandins are not secreted by prostate gland, but prostaglandins are secreted by seminal vesicle. Okay. Now, yeah, secreted by seminal vesicle. Now I'll move to the next topic, that is external genitalia, which present in human male. We all know that external genitalia is actually the penis, right? Now this penis has the tip of that penis is known as glans penis. We have already seen in the first slide, and it is the skin which is present that is known as glans penis or prepuce, which is the most sensitive part. Now when we see the transverse section of external genitalia present in human male, then we can see that this consists of 3 cylindrical erectile tissues which are encased in a sheath, and this sheath you can see here is Buck's fascia. So this sheath, the inner sheath is deep fascia. This one, this is also known as, the sheath is also known as this one, Buck's fascia. Okay, now the 3 erectile tissues which are present, the group of tissues which are present, 2 are dorsolateral and 1 is ventral. So dorsolateral are corpora cavernosa. It's written here. 2 are dorsolateral. This is dorsal side, this is ventral side. It's written here, dorsal and ventral. So here you can see that these 2 are, this one is ventral side, this is single, smaller, that is corpus spongiosum, and here are the 2 here, and that side, the opposite side, that are the 2 corpus cavernosa. So corpus cavernosa 2 in number, corpus spongiosum which is ventral 1 in number. In the middle, the artery supply, that is the blood supply, is there, and they are actually these they together known as corpus cavernosum of the penis. So all the at the extreme, like the end where the urethra is present, which is actually meant for ejection of semen into the female reproductive tract, right? And we will see now that what are the role of these 3 erectile tissue, because they help in erection of external genitalia for ejaculation of semen inside the female reproductive tract. So we have already seen that these 2 are the corpus cavernosa and this is the corpus spongiosum, and their function is penile erection. Actually what happens, once to achieve the erection, the brain sends impulse to the nerve in the penis, and that cause muscles around the corpus penis to relax. Muscles relax. The muscles which are present around here, that is, you can see here the deep fascia, that is Buck's fascia, and here is the superficial fascia. So these are the dartos muscle which are already present. We have seen in the previous slide dartos muscle which are present in the spermatic cord. Okay, inside that the smooth muscles which are present. So at the signal which they have got from brain, these muscles around the corpus cavernosum, they relax. When they relax, this allow blood to flow in the open spaces which are present here in these 3. Now when the blood flows inside, that blood creates pressure, making the external genitalia to expand. Okay, when it expands, then it allow the passage of semen inside the female reproductive tract, and once the muscles again contract, then the erection is reversed. So these 3 play important role in ejection of semen. Right. Now we'll move forward to the important process, which is actually point of your board point of view, which is very very important. That is spermatogenesis. Now we all know that spermatogenesis means the process of formation of sperms within the seminiferous tubules. We have seen the structure of seminiferous tubule. This is the testis, and here is the testicular lobules. We have already seen these structures. These are the seminal vesicles, and inside the seminal vesicle, if you have if you have the idea of the previous slide, that seminal vesicle has the basement membrane, after that germinal epithelium, and that germinal epithelium has the spermatogonia, and the spermatogonia, they are meant to produce sperms. So the developmental stages we have seen in the previous slide. Okay, so the sperms are produced inside the seminiferous tubules by the process we all know, that is meiosis. So first here I should start that actually at the embryo stage, the primordial germ cells, they developed inside the fetus from extra embryonic mesoderm. So the germinal cells, germinal epithelium, which form the sperms, okay, they are actually derived from primordial germ cells, and these primordial germ cells are actually formed from extra embryonic mesoderm. It is not from the embryonic mesoderm, but it is extra embryonic mesoderm. What is extra embryonic mesoderm? We will see in the coming slides in the fetal development. So extra embryonic mesoderm, they will differentiate primordial germ cells. These germ cells, they will migrate from extra embryonic membrane to the yolk sac. Okay, so they will come to the yolk sac, because in the embryo, first the yolk sac was present initially, but this yolk sac will later on, it will destroyed in human embryo, but initially it is present. This yolk sac is also a type of extra embryonic membrane, but it will become useless in human male in the later stages. So this extra embryonic mesoderm will carry the primary the primordial germinal epithelium, then the germinal cells. So these germinal cells will be carried to the yolk sac. From yolk sac it will reach to the gonads, which will be of course formed in the embryo, growing embryo. So extra embryonic origin is actually the primordial germinal cells. That's important for the point of competitive exam NEET, right? Now we will see the process that I should start, that at the onset of puberty, in male the puberty starts at 13 to 14 years. So at the onset of puberty, the germinal epithelium, they will undergo mitotic division. From mitotic division, they will form so it is actually the first phase. I should write simultaneously the different phases also. This is multiplication phase, where the spermatogonia multiply and form so many spermatogonia. Multiplication phase. So these spermatogonia will become more in number, many spermatogonia. Of course, because they are formed from germinal epithelium, so they are diploid. Now one spermatogonia will undergo the second phase, that is the growth phase, and it will increase in size. So the growth phase will result in differentiation of primary spermatocyte. Only the growth occurs. So the ploidy remains same, that is diploid. Primary spermatocyte now undergoes differentiation, and this is actually the first differentiation, from where the meiosis starts. So the first meiosis, and we all know that first meiotic division will result in 2 cells, and these 2 cells are actually secondary spermatocyte. The secondary spermatocyte, first meiotic division will be the reductional one, right? So when it is the reductional division occurs, then the ploidy will be reduced, it will come to the half. So the secondary spermatocyte will n in number. So 2 secondary spermatocyte. Now the second meiotic division. So when the second meiotic division occurs, then each secondary spermatocyte will form spermatids. But the second meiotic division is equational one. So the ploidy will be n, 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 and n. So how many spermatids are formed? 4 in number. 1 spermatogonia, 4 spermatids. This is first differentiation will form spermatids. So spermatid are just round structure, round cells, right? Now, but we have seen the, when we see the diagram of spermatozoa or sperms, then this sperm has the head, of course the tip of that is known as acrosome, then the neck and the middle piece, and then the tail. So now the spermatids have to get differentiated. So the first differentiation, now the second differentiation. The second differentiation where the spermatid get differentiated into spermatozoa. So all spermatid will result in spermatozoa. Of course, the one spermatid will result in one spermatozoa, so 4 will form 4 sperms. And the second differentiation which is occurring here, this is actually known as a specific term which is used for the differentiation of spermatid to sperms are spermiogenesis. Spermiogenesis. Okay. So spermatogenesis means from spermatogonia 4 sperms are formed. Spermiogenesis means from 4 spermatid sperms get differentiated. So you can see in this diagram, spermatogenesis, testis, spermatogonia, 2n, 46, 2n, 46, 23, n, 2 division, first differentiation, second differentiation, spermatid forms the sperms, spermiogenesis. Okay, so spermatogenesis is the process where the spermatogonia forms 4 sperms, first through mitosis and then meiotic division. Okay, yeah, this is the closed, very close view of the complete seminiferous tubule, where you can see spermatogonia 2n, then spermatocyte 2n, then secondary spermatocyte n, because here the first meiotic division, then spermatid, then spermatozoa. And look at these sperms. This is the Sertoli cell, large cell which is present in between the spermatogonia. That is a large Sertoli cell which provide the nutrition to the sperms. So their head is embedded before release. Their head is embedded inside that Sertoli cell so that they can get the nutrition. Okay, so that's all for spermatogenesis. Okay, the spermatogonial cell has 46 chromosomes in human male. Give the number of chromosomes in primary spermatocyte and spermatid. One by one we will take. Spermatogonia 2n. Human chromosomes 46, 23 pairs, 2n. Right? Now primary spermatocyte. We have just now seen that in the gonia 2n and primary spermatocyte is also 2n. It is just the growth phase which will result in primary spermatocyte from spermatogonia. There is no division, only the growth phase. So the primary spermatocyte has also will be having the 46 chromosome only. And what is the spermatid? After meiosis, it will form secondary spermatocyte, and secondary spermatocyte will form spermatid. So from primary spermatocyte, when the secondary spermatocytes are formed, then N, 2 cells, and from secondary spermatocytes, spermatids are formed, and these spermatids are also N. So when N means half of the number, so 46 half, 23. These are actually the sperms only, but spermatid gets transformed into sperms. Clear? Okay. So I think the concepts are now clear for spermatogenesis. Now we'll come to the another important concept, 3-mark question. Hormonal regulation of spermatogenesis, that is formation of spermatid, after that formation of sperms. And spermiogenesis means differentiation of spermatid from sperms. So hormonal regulation, hormones right away from hypothalamus. Now hypothalamus which secrete GnRH, gonadotropin GN, RH releasing hormone. We all know that hypothalamus secreting releasing an inhibitory hormone, so that when it releasing hormone, it will stimulate pituitary gland to release its hormone. When it secretes inhibitory hormone, then it inhibits pituitary gland to inhibit its hormone secretion. So it will secrete GnRH. Now this GnRH will stimulate pituitary gland to secrete the hormone. GnRH will act on pituitary gland, and the pituitary gland will secrete first FSH and second LH. So pituitary gland will secrete gonadotropin. So their name is gonadotropin. So gonadotropins are released by pituitary gland, and gonadotropin releasing hormone are secreted by hypothalamus. Okay. When FSH and LH are secreted, of course they will act on gonads. So the gonads in male is testis, right? Now in testis, the seminiferous tubules are also there, and seminiferous tubules, between two tubules the interstitial fluid is also there. So the different type of cells which we have studied in the previous slides are, we have seen that the 3 types of cells. Sertoli cell, just now we have seen that Sertoli cells provide the nutrition. Okay, so FSH will act on Sertoli cell, and this Sertoli cell will stimulate spermatogonia for spermatogenesis, first factor. And the second one, spermatogonia, it will directly act on spermatogonia. So it will undergo spermatogenesis. And Sertoli cell also secrete inhibin. We have seen in the function of Sertoli cell, it secretes androgen binding protein, it provides nutrition, it helps, it stimulates spermatogonia for spermatogenesis, and it also provides inhibin. This inhibin has the negative effect on pituitary for secretion of FSH. In case the sperms are more in number and not required anymore, then inhibin is secreted, so that FSH will not be secreted. If FSH will not be secreted, then spermatogenesis will be stopped. Now very important one, the LH. LH has the effect on Leydig cells. If you remember, outside the seminiferous tubules, Leydig cells were present. Their function is to secrete testosterone. And the primary function of testosterone is, first one, that is secondary sexual character, and also spermatogenesis. This spermatogenesis, this testosterone which is secreted by Leydig cell, it has also the paracrine effect on spermatogonia. When the testosterone is secreted, then only the spermatogonia will start forming sperms at the onset of puberty. So Leydig cells or testosterone, and Leydig cells will work under LH. So LH is more important for spermatogenesis. See, spermatogenesis is done by spermatogonia, but spermatogonia will undergo spermatogenesis in presence of testosterone after puberty, and this testosterone is under control of LH. This LH is secreted by pituitary gland. Now, this testosterone which is secreted, it also has the negative effect, and more important, it has the negative effect on both GnRH and gonadotropin which is released by pituitary. So hypothalamus पे भी negative effect है, and the negative effect on LH also. This is actually the feedback mechanism, that GnRH will secrete FSH and LH, they will stimulate spermatogenesis, but again there are Sertoli cells and testosterone which have the negative effect. So there is the hormonal system in our body, endocrine system in our body, it work under feedback mechanism. So hormonal regulation, yeah, this is the complete diagram which we have already studied. Gonadotropin, GnRH, FSH and LH. FSH, Sertoli cells stimulate spermatogenesis by spermatogonia, and then the inhibin which has a negative effect on FSH. Now the LH which have the effect on Leydig cells, testosterone is secreted. Testosterone has the negative effect both on LH as well as GnRH. So two important concepts we have seen: spermatogenesis and hormonal regulation. That's all for today, children, and in the next coming slide we will start with the female reproductive system, its various parts, and the oogenesis process and hormonal regulation. Thank you.